And we're very excited to have one of our Michelson Award winners, Ansuman Satpathy, who is instructor in the Department of Pathology and Postdoctoral Fellow in the Department of Genetics at Stanford University, and his research focuses on developing new genome sequencing technologies to study immune system function and dysfunction in patients. He has been working hard in this arena, and we expect great things from you going forward, but he's going to take just a little bit of time to talk to us about his work.
So thanks very much, and thanks to the Foundation and the Human Vaccines Project. I'm really delighted to be here. So as Peggy said, my name is Ansuman Satpathy. I'm an instructor in pathology and also a postdoctoral fellow in the Department of Genetics. I'm working with Howard Chang and Will Greenleaf at Stanford. And I'll talk about something a little bit different. What we've been doing basically over the last few years to develop new epigenome technologies to study primary human immune cells. And I'll tell you what I mean by that. So what our group is generally interested in understanding is how regions of non-protein coding DNA in the genome impact the expression of disease-associated genes. Okay? So this is the 98% of the genome that we often don't hear about, we often don't study, right? And so when we talk about this non-coding genome, we're primarily talking about two things. We're talking about cis regulatory elements, which are typically thought of as enhancer regions that control gene expression. And we're talking about trans inputs, which are typically thought of as transcription factors that bind to these elements and bring them in close proximity with the genes that they regulate. Okay, so why do we care about this? So other than the fact that it's the majority of the genome, studies over the last 10 years or so have shown, for example, genome-wide association studies, that the majority of disease risk actually falls into this area, right? So across many, many diseases. In this case, I'm showing an example from autoimmune diseases where about 90 or 95% of the risk actually falls into this non-coding space, right? Only 5% falls into the protein coding exome. So clearly there's something important to understand here, and it's important to measure that directly in patients and directly in the setting of disease. Okay, so how do we do this? And there are essentially two challenges here. The first is a linear challenge, which is as you walk down the chromosome in the genome, where precisely the bases that are open, active, and regulating genes. Okay? So where are the sites that are regulatory? The second challenge is a more complicated challenge, a three-dimensional challenge. So even if you know where those enhancer regions are in the genome, they could be hundreds of kilobases or megabases away from the genes that they regulate. So how do you now in three-dimensional space, in the folding of the nuclear chromatin, understand which cis regulatory elements binds to which gene? Okay? So the problem has been for immunologists that prior gold standard methods in the field have really required a large number of cells to do this. So 10, 100 million cells, even billions of cells to do this. So you really could never take these measurements in specific immune cell subtypes, certainly not from humans, and certainly not from patient samples in a serial fashion, right? So you really never had access to this 98% of the genome that's controlling genes. So what our group has done in the past few years is develop two new technologies to address these two challenges. Okay? The first is a method called ATAC-seq, or Assay for Transposase Accessible Chromatin with Sequencing. This is a method that was developed by a former graduate student in the group, Jason Boyne-Rostro. And what Jason figured out is that you could take this naturally occurring bacterial transposase called TN5, whose function it is to just cut and paste DNA across bacterial strains. Okay? So it mediates the hopping of transposable elements. What Jason did very cleverly is that he just incubated this enzyme with Illumina sequencing adapters. Okay? Then you take that complex and you just dump it onto cells or nuclei, and then it just naturally integrates these sequencing adapters into these open regions of chromatin, which are active regulatory elements. Then you can amplify those regions by PCR and then really identify with base pair resolution as you walk down the genome where are the regulatory sites in the genome. Okay? So you can find them with really high precision. And I think this has really been a revolution for immunologists because you can now do this in primary cells that you care about and that we study every day, right? So initially, you could do this in 50,000 cells. As I'll show you later, you can now do it in single cells. The problem, however, is that while it's really good at finding these sites, you can't link them to the genes that they regulate. Okay? So what I'll tell you about in this first part of the talk is a new method that we've developed called H3K27 acetyl high chip. this method now allows you in primary cells to identify functional enhancer promoter connections genome-wide using a single assay. Okay, and I'll just briefly walk you through the details of the method. Essentially what we do is in situ, we cross-link the chromatin architecture, so we cross-link the 3D contacts, and then actually physically ligate them together using a DNA ligation step. And then we pull down, using a chip protocol, just the regions that are decorated by this active chromatin mark called H3K27 acetyl. And then we make a library of these hybrid fragments. Okay, so the fragments are made up of 2 pieces of DNA, which could have been close together in the linear genome, or they could be far apart, but they're all acting together in the 3D nucleus. Okay, then we sequence to then infer the contacts between those sites. Okay, so let me walk you through some of the primary data. So this is a high-chip experiment done in K562 acute myeloid leukemia cells. This is a cell line. And what we're going to do is just focus now on one locus, one position on the chromosome, and that's the promoter for the gene MYC. It's a common oncogene. And so what I'm doing here is just setting our anchor point of analysis at that MYC promoter, and then we're just walking down the genome and walking down the chromosome and asking, where do we see high-chip signal? Okay, so what regions are making contact with this promoter? So you can see we get 5 sites, 3 that are relatively close by, within 200 kilobases or so, and then 2 that are actually quite far away, 2 megabases away from the promoter, and actually hop over 2 other genes to contact MYC. Okay? So this is nice, but you get a lot of noise with these types of assays. So how do we actually know the signal that we're getting is some type of functional enhancer interaction? So what we did in this case and several other cases is to compare our data to high-resolution CRISPR-I screens that were really coming out almost every day at that time. And here I'm showing you a comparison to a CRISPR-I screen done by Eric Landert and Jesse Engritz at the Broad Institute. So what Jesse and Eric did is they tiled 100,000 CRISPR guides along this exact same locus, okay, and then asked which of those CRISPR guides impacted MYC. So this is a really beautiful and elegant experiment. Took a lot of time to do, right? And they find 5 sites which perfectly overlap with the 5 sites that we find. Right? So we can recapitulate this 100,000 CRISPR guide experiment using a single assay. So I'm showing you, of course, just one gene locus here. We get the same information for every gene genome-wide using a single assay. Okay, so someone could say, do you actually need to do this type of high-resolution, precision, 3-dimensional analysis in every cell type? Immune cells, maybe they're relatively similar and they have an invariant chromatin structure. So the enhance, you could do it once and then predict it from that. So now what I'm showing you is the exact same MYC locus, okay, but now done in 2 different cell lines, a T cell line, myeloid cells, and a B cell line, GM12878 cells. And what you can see is a dramatically different enhancer landscape in these relatively similar cells. Of course, they're not similar to this audience, but they're both lymphocytes, okay, compared to a skin cell or a heart cell. They're relatively similar. Even down to the direction in which the enhancers go, right? So so T cell enhancers go off to the right here, B cell enhancers off to the left. We can then validate these results using our own CRISPR-I experiments and show if we knock down B cell enhancers in B cells, you lose MYC expression. Not so if you do it with the T cell enhancers. Okay, so can we actually now do these measurements in primary cells? This was the whole point of developing this assay. And so just to set up the challenge for you, the challenge is to improve the signal to noise of this type of assay by about 3 orders of magnitude. Okay, so from 50 million cells, which is the prior gold standard, to 50,000 cells. So what I'm showing you now is 3 high-chip experiments in 3 starting cell pools, 25 million cells, 1 million cells, and 50,000 cells. And we use this as sort of a benchmark for what's getting in the realm of primary cells. And the plots are these 2D interaction maps. So this is the raw high-chip data. And what you're looking at is you're walking down the chromosome and asking what part of the chromosome does each piece make contacts with. So the large diagonal is each piece of DNA interacting with itself. Anything off the diagonal, like this dot here, is 2 pieces of DNA that are far apart coming together to make a chromatin loop. So you can see we get nice signal with 25 million cells and then maintain that in 50,000 cells. Okay, so at that point, we moved to primary immune cells, and we moved to our favorite cell type, which is CD4 T cells, recruited 3 healthy volunteers, and then from a single blood draw, isolated, sorted out naive T cells, T helper 17 cells, or T regulatory cells, and then did high chip. Okay, and so what you can see is these really beautiful chromosome conformation maps, And what you're looking at as you go left to right is essentially zooming in to a piece of a chromosome. Okay, so you can see higher order chromatin structures on the left, and then zoom in to sort of chromatin domains, and then individual interactions between a gene promoter and a distal enhancer. So the first question we asked when we had this primary data is how do the interactions look? Do we get complex interactions, or could we have just predicted it from, the nearest enhancer goes to the nearest gene, and so forth? But what we actually see is a lot of complex interactions. I'm showing you 4 examples of that. So overall, about 80% of the interactions that we see are not just simply nearest enhancer to nearest promoter. Okay, so we see examples of enhancer-promoter skipping, which means that a distal enhancer will skip over intervening genes to target the expression of a distal gene. We see several enhancers working together often to control gene expression. We see promoter-promoter interactions, so promoters acting as enhancers for other genes. And we see enhancer-promoter switching, which is essentially dynamic enhancer usage for a single gene. And I don't have to tell this audience, as we've heard several times today, that the information that you get from doing it in a primary cell type, a sorted cell subset, is much more useful than what you can get from reference maps or bulk cell lines. So the genes that were Treg specific, we could see nice chromatin structure. You totally lose it if you do total T cells. You lose the sensitivity, and especially if you do cell lines. So you really need to do the assay in the primary cells that you care about and in human cells to get the right information. So I'll just end this part by returning to the original question that I posed. Can we use this type of 3D chromatin structure to predict or potentially nominate target genes of intergenic SNPs? Okay, so we focused on autoimmune disease because we have T cell maps. And I'll show you 2 specific examples first, and then we'll zoom out to more global analysis. So if you just focus on this bottom left plot here, what I'm doing is setting the analysis now instead of at a gene, at a SNP of interest. Okay, so this is a SNP that's associated with type 1 diabetes and rheumatoid arthritis. It's been validated several times in multiple studies. And that SNP is actually closest to the gene SMIM20, which when I saw that, and I'm sure most of you, doesn't really ring any bells. It doesn't have really any known immune cell function. But of course, that was a nearest gene, so that's what the studies had predicted as the target. But you can see that we really don't find any high CHIP signal at all at that gene. But instead, we see really nice high CHIP signal at RBPJ, which is a Notch regulator known to function T cells, and STIM2, which is a T cell activation gene. Same thing if we look at a SNP associated with MS, closest to the gene RMI2, but actually has very nice interactions with SOX1 and CLEC16A, which are also been shown to be involved in autoimmune disease. Okay, so if we zoom out now and do this for about 21 autoimmune diseases where we have really nice GWAS data, we can expand the list of nominated target genes by about 4-fold. Okay? And only about 50% of the nearest genes to those SNPs had any signal at all in our 3D chromatin conformation maps. We can go a step further and ask, how does the risk SNP actually affect the chromatin loop? Does it strengthen the loop, making gene expression stronger, or break the loop? And you can see we get dots on either side, meaning that it does both things. So as we move forward now into thinking about how can we use this information to target genes and target cellular therapies, we're taking both approaches. So we're trying to strengthen enhancer-promoter contacts using CRISPR or break them. Right? So on the left here, now I'm showing you those same 2 SNPs that were associated with disease that we just talked about. But now what we're doing is we're targeting that enhancer with a CRISPR interference, a dead Cas9 and Crab domain containing complex. So we're bringing a repression domain to that enhancer. And what I'm showing you is that we can now dial down the expression of the genes that we predict by high CHIP. So this validates the approach. This validates the data that I've just showed you, but also maybe nominates ways that we could use that information to target cell therapies. And conversely, if we target CRISPR activation domains to these enhancers, so these are 4 enhancers for the T cell gene CD69, you can now dial up the gene expressions of the genes that you predict by high CHIP, both by RNA and protein. Okay, so in the last few slides, I want to tell you sort of the next level of that. So it's very nice to have this epigenomic information, understand what are the molecular switches in the non-coding genome, but of course, everybody here is probably thinking it's not enough to do it in 50,000 cells. You need to go down to single cells, and we know a lot of things about T cells already, so why lose all that information just to gain epigenetic information? So we'd really like to be able to pair these pieces of information with single cell resolution, right? So get the epigenomic information, particularly for T cells, get TCR sequence, and then maybe even add additional layers, right? Transcriptomes, proteins, cell surface proteins, and other things. So in the same way that maybe Google Maps is useful because you have many layers of information, you know where the streets are, where the people are, where the stores are, you can use that to navigate the map, right? In the same way, we want, epigenetic information is important. It tells you a lot about the chromatin state, but we want to integrate that with all these other pieces of information. So just in a few slides, I'll show you. So this is all work in collaboration with Mark's group and Naresha Salagrama, who's a really fantastic postdoc in his group. So what we've been able to do is do exactly that. So take now this epigenetic profiling to single cell resolution, and then pair that, in this case, with TCR alpha and beta sequence from every single cell. Okay, and I'll just briefly walk you through this. So essentially what we've done is miniaturize the whole protocol for ATAC-seq in a microfluidic device. So we've engineered the device to do specific chemical processes in sequence. So the cells are individually captured, then go through a series of ATAC-seq steps by just serial addition of different chemicals. And then we targeted amplify the TCR alpha and beta locus or RNA from each cell, and then pool everything together, amplify together, and then barcode the libraries and sequence, and then computationally integrate TCR and ATAC-seq profiles for every cell. Okay, so here's what the data looks like. So what you're looking at here is chromatin accessibility in a piece of a chromosome. This contains the CD4 gene. And on top, you have reference data from the prior gold standard method called DNase hypersensitivity sequencing from 10 million cells. The red line is ATAC-seq data from 50,000 cells. And the blue line is now single cell ATAC-seq data from about 200 cells aggregated. So you can see we can recapitulate the information in 10 million cells now in 200 cells. But of course, now we have that information for every single cell, right? So as the rows in this heat map are individual cells, and the columns are individual enhancers and promoters. And so you can see things like enhancers that are very constant across T cells and some that are variable. And then, of course, for every single cell, we also have TCR alpha and beta sequence. Okay, so what does this data look like? This is a reduced dimensionality plot, a t-SNE plot on primary T cells now, so sorted in the same way as in the high chip experiment. We sorted either naive T cells, bulk sort of memory CD4 T cells, or T helper 17 cells. And they largely cluster by how we sorted them with some important differences. There are some naive cells that actually cluster like memory cells. And then there's some variation in the T memory population. So how do we get to this plot? So for every single cell, so here's a naive cell. The cell that was sorted is a naive cell, but actually clusters with memory or T helper 17 cells. We can zoom in and look at the cis regulatory elements. But we can actually infer the activity of transcription factors by diving deep into the sequence of each enhancer. And that's because you can think of it as you're spray painting a wall. So, right? So if you're spray painting a wall and you put your hand in front of it, when you're done, there's an impression of your hand, right? And in the same way, when you're spray painting the genome with transposase in ATAC-seq, if there's a transcription factor sitting down on the piece of DNA, it'll leave an imprint there. And so we can use that imprint to then infer what transcription factor is sitting at a particular site, do that over and over and over for every peak to get sort of general activities of transcription factors genome-wide for every single cell. And so in these bottom plots now, what we've done is paint the same t-SNE plot, but now from blue to red with the activity of a number of transcription factors. So you see things that make sense, like ROR gamma T being very active in a lot of T helper 17 cells, but not all of them. And then you can find new transcription factors by doing this over about 3,000 different motifs. Okay, so this is the last data slide. So I just want to show you one example of how we've been able to do this in a different setting. This is a patient who had metastatic basal cell carcinoma and was being treated with immunotherapy, was working really, really well for the first 6 months. Something happened at 6 months and stopped responding. So we thought, why don't we go dive deep into that person's T cells and see if we can figure out some chromatin state changes that may have happened that reflect that change in response. So this is now a reduced dimensionality plot from about 6,000 cells. This is not our single-cell RNA-seq. This is now single-cell ATAC-seq data. Okay, and so for the T cell cluster, so you can see a large number of tumor cells, and these are some stromal cells here, but you see a ton of T cells. And the dark dots represent the biggest clone in that T cell population. And so now what we can do is zoom into that clone and then compare the clonal cells versus all of the bystander cells. And you can look at the activity of transcription factors overall in that clonal cell versus bystander cells, and then zoom in to individual enhancers to identify potentially dysfunction-associated enhancers, for example, next to CTLA-4. Okay, so what I've told you today, I'm sorry that was very quick, so I apologize for that, but hopefully what I've been able to convince you of is that we're really working hard to develop new technologies to study this non-coding space in primary immune cells. We really want to be able to take these measurements in human cells. So we've done it in T cells because that's what we're most interested in, but of course this should be applicable to any cell type or disease process that you're interested in. And one thing that we've learned is that it's really important to do it in the primary cell type, right? You don't get the same information if you use reference maps, certainly not from cell lines, not from ensemble populations, and for the specific sequences, you also don't get the information from mice. And as we move forward, we're thinking about, okay, we can read this information really well now, but can we actually use that to engineer cells to do new functions, make them more durable, long-lasting, things like that. So again, thanks to the Human Vaccines Project. It's really great to meet you all and become part of this community. We weren't working on vaccines before, and I think this will now allow us to move into that area. And we'd like to use all of these approaches to really study why vaccines work well in some patients and not in others. And hopefully the next time I see you all, I'll have some preliminary results for that. But we're really working on the whole gamut of sort of epigenomics, 3D measurement tools, single-cell epigenomics, pairing that data with orthogonal information, TCR, BCR, even CRISPR guide for high-throughput perturbation screens. And we're also working very hard on the computational and statistical analysis side. So thanks very much. Just thank a bunch of people here, in particular Max Mumbach and Naresha, who were really the trainees that worked very closely with me on this. Thank you.
Okay. Well, thank you very much. Very impressive and sophisticated work.